Welcome to 12-minute summary of 12 years of research on bioboundaries and why they're our best chance for stopping wild dogs from going extinct. None of you will be unfamiliar with this. We all know that habitat loss and the associated human-wildlife conflict is the biggest threat to predator populations of all species all over the world. And particularly for the African wild dog, with an endangered classification, probably only 700 packs as breeding population, and humans as the main cause of death in all of the populations, it is certainly something that needs urgent attention. It's worthwhile to remember that 95% of African wild dogs live in open systems. They're not fenced in. They're only separated from livestock, which is how they get into conflict, by fences which are very often knocked down by elephants or burrowed under by aardvarks and warthogs. So African wild dog bioboundaries, the idea is to keep them inside the safety of protected wildlife areas. As long as they're inside there, they are reasonably safe from human-wildlife conflict. The concept of a bioboundary is based on mammals communicating via odours in various body secretions, urine, feces and scent marks, and particularly that carnivores mark their home range with scent marks. And the plan is to make artificial equivalents of those scent marks that send the same keep out messages as the natural ones, and use those to keep the wild dogs inside the protected areas. So we need to formulate artificial wild dog scent marks, and the practical problem that we face is answering the question what chemicals in the real wild dog territorial scent marks send the keep out signal. The process goes as a cycle. We need an understanding of the biology of the scent marking, who scent marks where, what the responses of the various dogs are. That informs which materials we need to sample in order to be analysing scent marks. The analytical chemistry gives us results in terms of the identity of candidate compounds that will go into the artificial scent marks. They are either produced by synthetic chemistry or bought off the shelf. Artificial scent marks are formulated, put back out into the field under conditions where we have known expectations for the responses of the dogs. The bioassay results then feed back into development of more candidates and refinement of the formulations. As study area, Megan Classer will already have highlighted some of this. It's to the east of Maremi in northern Botswana. And as far as the sampling for the analytical chemistry goes, we collect urine, feces and other scent mark materials from known dogs, known dates, known locations, and we analyze them by gas chromatography mass spectrometry in the BioBoundary lab in Maui. That presents a substantial analytical challenge because any mammalian odour consists of several hundred different organic chemicals, and the challenge is to work out which of those are actually sending the signal that we're interested in. So in this case, the signal that says to neighbouring packs that they should not venture further because they're going into another pack's home range or territory. There are two approaches we can take. The first is called reverse engineering. The other one response guided or message guided strategy. Reverse engineering asks if we had to design a scent mark from scratch that would operate under the environmental conditions that we know exist in the field, what chemicals would we put in it to send the signals? And we know that anything would have to be airborne because the dogs detect it by scent. Obviously has to be persistent because the signal lasts for a couple of months. That implies chemical stability, and what we're in addition looking for is signs of biological adaptation, so something which is not just straightforward chemistry. So this is one example of reverse engineering. These two particular peaks on a chromatogram here. Quite interesting because two peaks means two different substances, but they've both got the same mass spectrum, which suggests the same structure. And if you look at the biology, there's also interesting differences between samples taken on subsequent days from the same dog. That points to some biological significance on its own. And by various chemical processes, those two peaks were both identified as 5-hydroxyhexanolide, which is of low enough molecular weight to be airborne, stable enough structure to be persistent, and it's got the footprint of biological adaptation on it because there are two optical isomers, two optically active carbon centers here, and that gives 4 enantiomers, which is a perfect solution to coming up with a stable scent code for coding something like pack identity. So those are good candidates for including in an artificial scent mark if we need to code pack identity. The message response guided strategy asks what compounds are present in materials which send signals compared to those which don't, and in materials which animals respond to versus those that they don't respond to. And that obviously implies that we have to know the biology of the scent marking, which materials carry the signal and how the animals respond to them. Otherwise, we don't even know what to analyze in the first place. We now know that urine and feces deposited at marking sites only stimulates any reaction from other dogs erratically and rather weakly. But in contrast, urine and feces deposited at shared marking sites, which Megan Classer told you about already, reliably elicits overmarking, a lot of sniffing, rub rolling from other dogs that encounter the scent. So we are absolutely sure that materials deposited at shared marking sites are scent marks, and if we compare those with the equivalent materials deposited at resting sites, that will give us clues as to what compounds in the material at the shared marking sites are sending the keep out messages. Now, we got a replacement GCMS for one that was destroyed by a power surge in June 21, and I pressed the reset button on the analytical work and redid analyses that had been done earlier, with a stress on comparing samples from marking sites versus material from resting sites. So it was a message response guided strategy. And this is, in very, very brief summary, is the kind of results we get. Identified this is an identity still to be confirmed. Everything else has been confirmed by co-injection with standards. And there are 5 compounds which will be incorporated into artificial scent marks and tested by putting them out at marking sites. So my next step, having identified those candidate compounds, is to formulate those into the artificial scent marks with, if necessary, some kind of controlled release carrier. And they will then be placed in these dispensers, which we've already developed. They will be put out at marking sites using a protocol that has already been validated, and we will record the dog's reactions. If the dogs react by overmarking, we know we've got a close approximation to a real scent mark, and that formulation will then be used to build a pilot scale BioBoundary. If the formulation doesn't work, there are other smaller and less well characterized components which will then be incorporated into the mixtures. So this is not a one-shot, it either works or it doesn't kind of exercise. This has obviously been a quite superficial, rushed summary of what we've been up to. There is various other things here, YouTube videos, etc., of wild dogs reacting to scent. And these 2 links at the bottom here are the full-length versions of what I've tried to tell you now. You'll be able to access these direct off the links in the video once that is loaded to the site. This is the huge number of people that has helped along the way. I'm not even going to try to list them because we don't have time. And these are our major funders and people who have helped us by sponsoring various materials and software along the way. I'm profoundly grateful to all of them, and I'm grateful to you for listening. Thank you, and I think there should be time for questions.